Good afternoon, everyone, and thank you for attending RS's webinar with Brian Canter on recapping the targeted radionuclide-based therapy workshop. Questions will be addressed at the end of the presentation with a Q&A, so please submit your questions using the chat box. Please note this session is being recorded. All registrants 2 to 3 business days via email. Moderating today is Dr. Jan Schumann. Dr. Schumann is a Radiation Research Society counselor and was a co-chair of this winter's workshop. He's a radiation biophysicist specializing in Monte Carlo simulation research for proton therapy and radiobiology. He is currently Associate Professor of Radiation Oncology at Massachusetts General Hospital and Harvard Medical School in Boston. As head of the multi-scale Monte Carlo modeling lab, his research focuses on improving clinical treatment through the use of Monte Carlo methods at multiple scales. Welcome, Dr. Schumann. I'll hand it over to you.
Thank you very much. It's my great pleasure to introduce this webinar. And first of all, I'd like to thank you all for joining. This is a presentation kind of recapping what we saw as the last meeting before the quarantine happened in many places. We had a few wonderful days at the Big Sur Resort with not a webinar. It's actually a real meeting back then with 4 topical sessions and 3 plenary keynote speakers on dosimetry and targets, radiolabeling, isotopes, radiation quality effects, theranostic companion diagnostics, and clinical implementation and translation of radionuclide therapy and the era of advanced radionuclide therapy, targeted radionuclide therapy. So this is part of kind of becoming an annual thing of the Radiation Research Society, where we hope to continue having winter workshops where we try to reach out or include topics that are often underrepresented in the Radiation Research Society annual meetings, and just kind of form expertise in these by inviting a lot of interesting people and hearing a lot about in-depth, really go into a topic. And this year it was radionuclide therapy. So we had a, I think, super interesting and fun seminar workshop. And so please look out for those. They're really fun. It's good if you can come next time. It's wonderful for both the location, well, 3 things: the location, the science, and really you meet a lot of people that are experts in these fields. So come next time. And Brian Canter came to this year's workshop. And so it's my great pleasure to introduce him. He participated, as I said, in the workshop. He's a PhD candidate at the New Jersey Medical Center, working with Roger Howell and studying the bystander effects of breast cancer patients with spread to the bone, looking at radium-223, which is an alpha emitter. He received a grant of the New Jersey Commission of Cancer Research, but mostly I know him because he is an active member of the SIT committee. He is representing biology, and he is very active, transformed the SIT newsletter and the annual workshop that is part of the Radiation Research Society annual meeting. So it's great to see SIT's so active, and he is also very passionate about science communication, where he strives to increase the trust between science community and the public by eventually aiming to have a career in science policy. If you want to see his work, he also directed, produced, and wrote a recent video that's published in of their work, published in the Journal of Nuclear Medicine. And yes, he's just a very fun guy to hang out with at the meetings. So also come to the meetings if you can. Just as a reminder, if you do have questions during the talk, we will answer questions at the end. You can type them at the chat window on the right side. I believe it's on the right side for everyone. And we will just address those questions once the presentation is finished. And with that, I'll give you Brian Canter.
Thank you, Jan, for the kind introduction. And I agree with everything you said about the workshop. It seemed like it was another sort of world ago, and it was less than 3 months. But I agree, the location was beautiful. That was evident from the sunset I saw on that first night. This talk that I'm giving today is just sort of a real broad overview of the workshop. As Jan indicated, I'm in the biology representation on the SIT committee. So some of the physics and chemistry was new for me when I was attending the workshop, but I thought that was one of the strengths of the program that was put together. And again, if you have any questions, please type them in the chat and they'll be answered at the end of the presentation. So there were a couple learning objectives that were listed with the workshop itself, and I kind of framed the presentation around these two objectives. And the first one being to understand the difficulties in implementing targeted radionuclide-based therapies. And then the second was to understand proposed ways for overcoming these difficulties. And then Dr. Schumann and Dr. Abergel, Rebecca Abergel, the other organizer, were nice enough to have the talks presented in a fashion that I think one built off of the other, and helped me really to form an outline and a spine for this presentation. So start with a brief introduction, then move on to dosimetry and targets and presentations around that, then to radiolabeling, isotopes, and radiation quality effects, then to Theranostics and companion diagnostics, followed by clinical implementation and translation, and then lastly, a few concluding thoughts to revisit some of those learning objectives. So the first evening of the workshop, former Radiation Research Society President Kevin Price gave what I termed a landscape evaluation of the field of the use of radionuclide in medicine, going all the way back to some of the initial work done many decades ago. He focused a lot of the presentation on alpha particle therapy, and in particular shared what I thought was a very representative figure of the increased clinical demand in the UK for radium-223, radium chloride. As Jan indicated, that's the compound that I'm working on with Roger Howell in my thesis research, but it demonstrates that after the approval of radium-223, there's an ever-increasing number of patients that are being prescribed, and in addition, the number of treatments as well is increasing. And then Dr. Price was nice enough in his talk to go through a lot of sort of the different challenges that then some of the other presenters touched on in their remarks as well, but mainly looking at the tumor geometry in biology, microdosimetry, the biological mechanisms that were responsible for some of the reductions in tumor burdens or slowing down of cancer in patients, the different tumor models that can be done, particularly animal models, but also in silico models, and then combinatorial approaches to combining targeted radionuclide therapy with immunotherapy or with chemotherapy, which some of the presenters went into more detail. The first topic, subtopic, dosimetry and targets. So the very first presenter on the next morning was Dr. Manuel Bardiez, and I borrowed one of his a cartoon that he had kind of created to demonstrate that with targeted radionuclide therapy, you're going for a systemic radiation treatment, so not focusing necessarily on just one particular organ or one particular tissue. And a lot of the way clinical trials are designed is around how chemotherapy is used with radiation, or chemotherapy is used in the clinic, and that can lead to underdosing of patients. And it's important to consider not just toxicity when doing dosing, but also efficacy. And so the way to be able to measure that dose that is reaching the target is through dosimetry, and he emphasized that how essential it was for dosimetry to be a calculated part of all of these clinical studies, and had to be properly documented as well. And he mentioned kind of candidly sort of a disagreement that, at least in the European Nuclear Medicine Association, about those with a more of a clinical background or physicians versus those in the physics groups, having sort of different priorities when it came to dosimetry, and whether how much the prioritization of dosimetry, and that comes to physicists and working heavily on carefully done models, and clinicians having to worry about the ease of use of dosimetry with their treatments, with their patients in their studies. So I thought it was an interesting perspective as a biologist I didn't really consider in my own thesis research. Following Dr. Bardiaz's talk was a talk from Dr. Rob Hobbs about microdosimetry, so sort of small scale modeling for radiopharmaceutical therapy. And one of the big challenges with radiopharmaceutical therapy is the complexity of the treatment. It's heterogeneous, and then you have radioactive daughters that are potentially moving away and causing increased normal toxicity in untargeted areas. And he delved into sort of three different studies that his group and colleagues have been working on with actinium-225 and kidney toxicity associated with the radioactive daughter bismuth-213. He delved into toxicity around the salivary gland, as well with iodine-131, and then also with radium-223. Mentioned that the hematotoxicity, the toxicity in the blood, was higher than what had sort of been broadly calculated in some of the trials. And the figure on the right is one I've done on my own to demonstrate that radium localizes preferentially to the inner surface of to the mineralized region of the bone, but the alpha particles themselves have the potential to delve into the marrow space, causing toxicity. And so some of the approaches that Dr. Hobbs suggested for improving the microdosimetry was looking at larger scale animal models, which we'll talk about in a few minutes, and then also having a relative biological effectiveness data for specific sub-organ sites, so the tubules of the kidney, as an example, or a portion of the cortical bone of the mineralized bone, rather than focusing on the entire bone itself. That was talk was followed by Dr. Marika Nestor, who looked at presented on radiosensitization strategies, in terms of improving molecular radiotherapy for cancer. And one of the approaches I thought was quite elegant was to ensure sort of the one of the hallmark tumor suppressor proteins, P53, can be stabilized through inhibiting one of its negative regulators, MDM2, which is visualized in one of the figures I took from her presentation, where a radionuclide conjugated to an antibody is targeting a cell surface marker, and then is going to inhibit MDM2, allowing for P53 to be allowing for the stabilization of wild type P53. Dr. Nestor also presented an additional strategy which targeted HSP90 with the inhibitor anolsipib. I think that's how you pronounce it, and that is to prevent DNA repair in the cancer cells, so with radiation therapy. And that was a similar approach with a different intracellular, in this case, target. But what it functioned to do is it allowed to overcome sometimes the ineffectiveness of molecular radiotherapy, and how it is quite heterogeneous, and that some cells don't respond and others do. If you can sensitize, use radiosensitors to make cancer cells more sensitive to the therapy, it could lead to a better outcome. And then in one of the last presentations on one of the final few presentations on this dosimetry topic was one of the abstracts from Alejandro Bertolet, who at the University of Pennsylvania, and was had identified that with alpha particle emitters, there are fewer interactions between the radiation and the matter, and the typical sort of dosimetry would not compensate as well for them. And so he built a site specific model for stochastic interactions, and found that basically by being able to model sort of all of the different activity in a particular sphere in a space, it was useful to predict biological effects such as cell killing. And then onto the second set of talks delved around radiolabeling, isotopes, and radiation quality effects. And the first of these talks really got to one of the approaches that Dr. Hobbs and really Dr. Price as well related in their talks, which are using larger animal models for evaluating nuclear imaging therapy. It was presented by Jeffrey Bryan of Missouri University, and he really presented the really great overview of the entire PET imaging center that's at the University of Missouri, and mentioned all the various different isotopes that were available, copper-64, yttrium-90, and also the number of different collaborators that went into helping run the different studies. So there were biochemists and chemists, and there were cardiologists and somebody looking at metabolism. And most importantly, what they were able to do is be able to model the radiopharmaceuticals in both dose and distribution, and relate that from dog to humans. So it was much more translatable than a smaller animal model such as a rat or mouse. And then following up on that, in that talk, Dr. Charles Maitz, also of Missouri, discussed the different choices for selecting isotopes for radiopharmaceuticals, and some of the challenges he highlighted in his talk, where he presented sort of several different studies that his group had worked on, were the retaining those radioactive daughters, the radiochemistry, when designing the radiation pharmaceutical, the process of making it, the synthesis, and then delivering, in the case of alpha particles, being able to deliver the alpha particles to the target. And he also outlined sort of the four broad categories of approaches: having elemental targeting, smaller molecules, like a peptide, and then bifunctional chelates, and with linker molecules, and then radio immunotherapy, so a combinatorial approach. And then I thought a really elegant presentation was done by Sarah Cheal of Memorial Sloan Kettering, talking about how to be able to reduce toxicity, or even the distribution of radiation in radioactivity in the bloodstream, and having more of it land in the tumor cell itself. And this was accomplished with a pre-targeting, with a bispecific antibody, so as it's sort of demonstrated in the figure, a bispecific antibody that recognizes both the tumor and also recognizes a chelating agent, DOTA, is injected, and then it deposits itself onto some of it will deposit onto the tumor, and some of this bispecific antibody remains in the blood vessel. But then, with the injection of a clearing agent that clears out all of this bispecific antibody from the bloodstream into the liver to be eliminated, and then once that's done, the DOTA target, the DOTA complex, so the chelator conjugated to the radionuclide, is injected, and some of it would be eliminated, but a lot of it will be able to go to the tumor, to the anti-DOTA portion of the bispecific antibody. And she highlighted some really great data and really great results that were used with a actinium-225 labeled alpha antibody, an alpha particle emitter, for treatment of HER2 positive breast cancer. And following Sarah's talk was a very nice presentation from Pierre Pouget, and Dr. Pouget really was trying to sort of understand kind of the mechanism for how targeted radionuclide therapy is going to affect cancer cells, and not only the targeted effects, what is typically thought of with the with damage to the nucleus, but also looking at the effect of, in this case, a radioimmunotherapy to the cell membrane, which created the formation of lipid rafts that, through some downstream cell signaling mediated by p38JNK, eventually led as well to targeted effects in other intracellular effects, but also to cytokine production that resulted in non-targeted cells undergoing a similar biological effect. And so I think it's a very important consideration to understand with targeted radionuclide therapy, it's not always the case that all of the cells, cancer cells, can be eliminated successfully with just the targeted effects. It is very much about understanding the contribution of the bystander effects towards eliminating the tumor cells, particularly in a metastatic disease. So Dr. Pouget's talk was followed by a talk from Yasser Gholami, who I think, along with his colleague from Australia, and I think there was another colleague from Australia, set the record for the furthest travel to Montana. And Yasser presented a paper or a presentation, and then had a poster as well that frankly went a little bit over my head initially, but it really, what he focused on was there are some of the challenges with chelation we'll get into a little bit in a few minutes, but rather than using a chelator, being able to use a nanoparticle delivery system with a radionuclide, and then using PET MR imaging, waiting specifically with and then waiting for specific values to be able to visualize that radionuclide in vivo, and sort of working towards, in the long run, a physics biomarker, so a biomarker that is dependent upon physically weighted physics properties. Following Yasser's talk, Jerry Nolan from the Argonne National Lab, the part of the National Isotope Development Center, gave an update on Argonne's production of astatine-211, which has a very short half-life, making it difficult of, I think, about 7 hours, which makes it difficult to deliver the astatine-211 a long way. And so, just in terms of the graphic on the right, kind of describes all of the different processes undergoing at the National Isotope Development Center. But Jerry Nolan talked a lot about the production and then purification of the astatine, now using a radon-211 isotope, with the ultimate goal of being able to extend the delivery range of astatine-211 to widen its use. And that concluded all of the sessions for the day. I decided not to include any slides of me skiing for the first time in 5 years that would have been highlighted by me attempting to put the downhill ski on first when my skis fell off after I wiped out on my second to last run, and being heckled mercilessly from the lift that I was supposed to do my uphill ski first. But nevertheless, I was physically a little bit tired, but I'm very excited for the second set of evening talks at the workshop. In the first talk was from Aron Popovtzer-alpha. It was about an alpha DART product using radium-224, and really focusing on how to deliver the radium-224 to the in this case to a squamous cell carcinoma in the head neck area, while reducing the amount of normal tissue toxicity. And so now this alpha DART product, you can kind of see from the figure on the right, using a CT-based treatment plan. There's about a 5 millimeter distance between these different between these seeds, kind of look like rods that sort of bore into the tumor, and then the radon is, or the radium is injected into through these seeds into the tumor. And there were some promising results that Aron presented from trials, and indicated that this was part of a larger sort of operation for the company with multiple cancer types, not just squamous cell carcinoma. But as an engineer by training in my undergrad, I really did appreciate the engineering feat. And then my mentor, Roger Howell, was fortunate enough to give the plenary talk, and as tempted as I was to include my own data as the representative slide from his talk, I instead chose to use what he did with this kind of the second half of his presentation, which focused on how to optimize really the radiopharmaceutical therapy. But he did start with a lot of the initial work done with iodine 131 that Dr. Price had presented as well, and then also with yttrium 90 and with Bexar, and sort of why they didn't succeed as well as they could have. And then Roger presented some of the work that our colleagues at Rutgers have done, looking at the effect of dose rate, because again, with a radionuclide, you have this chronic radiation, so dose rate is a consideration to take into, and then also the bystander effect, some of the work that I've done along with another graduate student in our lab, Calvin Long. But I thought Roger, we haven't done as much work on this lately, but the antibody cocktail that he presented as really trying to overcome the heterogeneity of the tumor surface cell markers. So just with the figure that Roger has, is ideally with a patient, you would be able to sample some of the tumor, and then be able to identify through flow cytometry the different cell surface, the different cell surface markers, and then the expression, the number of them. And so to indicate, for example, on one cell there might be 4 or 5 times the amount of them of target that there is on another cell. And so then being able to do in combination, and after the biopsy, being able to do a Monte Carlo simulation and even a survival analysis as well, can really help inform a treatment plan that would be patient specific. Then moving on to the third unit was focused on Theranostics and companion diagnostics. And the first talk of the morning was from Ravi Patel, and I thought what was interesting about Ravi's talk was how new he was to immunotherapy, but as a I believe a radiation oncologist, he really recognized that the being able to raise the therapeutic index or widen the amount of treatment going to the tumor cells versus going to the normal cells was a big challenge for metastatic cancer, and that immunotherapy agents, particularly the checkpoint inhibitors, have been are being approved regularly. And so he was able to combine his targeted radiopharmaceutical therapy with an yttrium radionuclide in combination with immunotherapy, both looking at a checkpoint inhibitor and then also a cytokine-based immunotherapy, and he found that the effects were dynamic, and they were also heavily dependent on dose. And then the next presentation was from Eric Price of Saskatchewan and the University of Saskatchewan. And for a biologist, this was maybe a flashback to some of the biochemistry that I took in my first year in graduate school, and then of course in undergrad. I think he, it was Eric did a great job of going through all the different radio metals for both imaging and for therapy, and then as well as all the different parameters that I've included on the right for chelators, for linking. They included thermodynamic stability and mechanics and pKa, and it really gave me sort of a better appreciation for how moldable one of these chelators are for a specific application. It's just really about identifying sort of what specific test is needed to validate the chelator. And then following Eric's talk was a discussion led by Catherine Vallis of Oxford, and it focused on combinatorial therapies of chemotherapy and radiation therapy, and the motivation being that there is significant toxicity associated with systemic chemoradiotherapy. And so Catherine presented a solution whereby using a indium, chelated indium radionuclide, as well as a chemotherapeutic similar to doxorubicin, and having them specifically transported to the tumor site. It was a way of lessening that toxicity, and in addition, the indium would be available as well to be imaged, and so it would give sort of better specific targeting with therapies that are more traditionally systemic. And then the next presentation was from Peter Kim, who was really trying to, for an individual patient that is having yttrium, the radionuclide yttrium 90 embolized and distributed into the in microspheres and then delivered to the liver. This was a way of being able to measure the the dose that actually was delivered to the different parts of the liver using an imaging-based method, in this case SPECT CT images. And really he focused on technical part about trying to identify the best way to self-calibrate the regions of interest to focus on for finding that heterogeneous distribution of the microspheres, and emphasize the need for that in the treatment planning. And then the last of the 4 topics was about clinical implementation and translation. And the first talk in this section was from Daniel Junot, and he gave a perspective as a clinician presenting a number of human trials with lutetium-177 and actinium-225, and in particular mentioning that lutetium-177 is approved for neuroendocrine tumors, but in the majority of the results he was presenting for both radionuclides were looking at their effectiveness against castrate-resistant metastatic prostate cancer, and the results that Daniel presented indicated both radionuclides were well tolerated for treatment, and were efficacious. There are, though, a number of challenges that he highlighted. One specifically that he focused on as a clinician was or two really, were the ease of use. So sort of how difficult, how invasive it was going to be, and then really dedicated a number of slides to the acceptance of the patient. And really what struck me was him mentioning that the patients were not necessarily scared themselves of the radiation, but they were more wondering if the fact that they were being injected with radioactivity, if that was going to affect their family and their friends. And it's also important to note that the supply is of these radionuclides, particularly for Actinium-225, is an issue. And so he mentioned this as a clinician, if the supply is there and it can be continuously reordered, and if the patients are willing to accept it, the doctors would use it in the clinic. And then the next presentation was from Olga Koper of Cardinal Health, which is a major distributor for radium-223 in North America. But she presented the business perspective, which can almost have a greater appreciation for in this post coronavirus world. Just really, she highlighted all these considerations to take in mind when adopting Theranostics. So the development, how the theranostic was going to be, or how the radiopharmaceutical was going to be manufactured, and then how well that could be scaled up to for increased demand. And then in particular as well, she mentioned having facilities that working with radiation, you had to have facilities that either that could be repaired pretty easily without someone having to don all sorts of PPE, but having proper personal protective equipment for workers was an absolute necessity. And then she delved into a lot about logistics and about ensuring that it was a robust supply chain, and that even something like the delivery vehicles were equipped properly for transport of the theranostic, and then making sure as well for the regulatory and quality assurance that the regulations vary depending upon jurisdiction, depending upon country or state, and then ensuring that the product would be ready to go to market on a specific timeline. So I thought it was a very different perspective, a non-scientific talk. And then that presentation was followed by Calvin Lung, who works with me at Rutgers, and as I mentioned previously, working with radionuclides, you have dose rate patterns that are sort of different than working with an external source. And Calvin's interests have really shifted towards how radiation affects the immune system, either stimulating or inhibiting. And so he looked at sort of three different markers, MHC class 1, calreticulin, and mica in human breast cancer cells after different irradiation patterns, which are highlighted in the figure, either an acute dose or a fractionated dose, or a chronic irradiation with an exponentially decreasing rate. And he saw marker specific changes. So the MHC class 1 differences across the treatment groups were different than the calreticulin, or were specific to MHC class 1 and different than the calreticulin, and then different than NICO. Then following Calvin's presentation, there was a talk from Amber Bates, who worked with Ravi on targeted radionuclide therapy combined with immunotherapy, and her motivation is really to understand sort of how the immune cells could better recognize the tumor cells, because that's a very big problem with immune cells not recognizing, or tumor cells escaping the immune system. And so her approach was kind of built towards using yttrium-86 radionuclide with an agonist of interleukin-2, and then along with a interleukin-2 receptor, and then along with a immune checkpoint inhibitor, and found that the effect with the immune checkpoint inhibitor was enhanced using the yttrium-86 and the IL-2 agonist, and it was actually very striking to see the mice that received all 3 treatments, all of them responded, all their tumors responded quite well in comparison to the other treatment groups. And then sort of rounding out the workshop was a presentation from Charles Kunos at from the National Cancer Institute, which is a part of NIH, and specifically is in the cancer therapy evaluation program. And he went through the entire portfolio of the radiopharmaceuticals, and I really encourage everyone to look through the cancer therapy evaluation program. He was very, very blunt in how much support he wanted to give it, a lot of support, any support that he could to investigators in the radiation research. But he really emphasized that we, as the radiation research community, needed to make the effort to work with him. And just the 2 graphs he sort of presented are that I thought were very striking. On the left, the sources of radiotherapy in their program now are radiopharmaceuticals only make up 2%, and the internal radiation sources are far outnumbered by the external radiation sources. But in the future, he anticipates, and his team anticipates, the internal radiation sources vastly outnumbering the external radiation sources, with almost 25% dedicated to radionuclide antibody conjugates. So presenting a large opportunity for the radiation research community to be one of the leaders in this field. And then the last presentation was from Dr. Scott Tagawa, who gave a clinician's perspective at Weill Cornell of having run a number of clinical trials with targeting PMSA, which is a verified cell surface target for prostate cancer. And two of the challenges he really highlighted were using PSMA as the target. Its expression varies on cells, so some cells may get hit, and some cells may not get the treatment, and some cells may avoid the treatment if they lack PSMA. And then delivering enough dose to actually hinder the cancer cells is another one of the challenges. But he presented a variety of clinical trials with a number of approaches, combinatorial therapies, both alpha and beta targeted radionuclide therapies, and then also antibodies and peptides used in the case of the alpha radionuclide therapy, in the case of what the radionuclides were conjugated to. So then just briefly, a few kind of concluding thoughts. I listed some of the difficulties that I thought the group really kind of helped identify for targeted radiopharmaceutical therapy: normal tissue toxicity, measuring the absorbed dose and all the complications with the dosimetry, delivering the drug to the target, off-target effects, whether they be bystander or abscopal, and then patient acceptance, really clinician acceptance, or even translating some of the research from animal models from the bench to the clinic. But I think through all these presentations, there are a lot of people working with wearing different hats and having multiple approaches to overcome these difficulties. And so I just want to acknowledge again Jan and Rebecca for organizing such a wonderful wonderful conference, and for being two very great people and really enjoyable to spend time with and to talk, and very supportive and positive of the SITs. And I want to thank the entire team at Meeting by Design for their help with the meeting and the webinar. Veronica, Audrey, Lindsay, Katie, and Jordan. I think I got everyone. I want to thank as well the Radiation Research Society Executive Committee led by Susan, the president, Susan Bailey, for setting up these webinars. And then I want to thank my colleagues at Rutgers, Roger and Calvin, as well as Edward Azam, who is on the executive committee and encouraged me to give this recap in this webinar. And thank you everyone for your attention, and we'll take questions.
Thank you, Mr. Canter. We have a few minutes for the Q&A, so I'll hand it over to Dr. Schumann to kind of give his comments and present those.
Thank you very much, Brian, for this really great recap, I think, of an entire 2.5-day workshop in more than 40 minutes. That's very hard. And I think you really nicely demonstrated how many different aspects really play together, and the problems of many of those different disciplines that are involved in radionuclide therapy. We actually have a few questions already. Ed Azam is saying, thanks, Brian. Were there any discussions on the suitability of organs on a chip to study radionuclide therapy studies?
Yeah, thanks for the question, Ed. I can get back to you on that, because we meet at least once a week. My remembering now, I don't recall if there was any specific strategies mentioned that included lab on a chip, but certainly that's a nice approach to be able to make a more sort of representative of model in vitro, and then also for high throughput to be able to run a number of different assays. I don't know, Jan, if you recall if there was anything mentioned in lab on the chip.
Yeah, it's as you said before, it feels like a lifetime away already. So I'd have to refresh my memory too. I feel like it was mentioned somewhere, but I don't recall exactly what the which presenter was talking about. Another question by Alan Harris, I think. Was PET-CT mentioned during the workshop?
Yes, Alan, thanks for the question. It was. Not trying to critique my presentation too hard, but I think it was much. I didn't focus as much on the imaging kind of portion of the talks, but certainly PET-CT was included as considerations for choosing radionuclides, those that were going to be used for imaging for that particular modality. That's a little bit sort of outside of what I do with my research. probably even a better question for Jan, or even my boss Roger. But Jan, if you recall any particular mention of PET-CT.
Yeah, I mean, we definitely it was definitely talked about also for just trying to get a better understanding of where the pharmaceuticals actually end up. So it's definitely part of a lot of research in trying to do symmetry and modeling of the effects that are being investigated. Similar as for the last question, I remember that we talked about it. I don't exactly know the details anymore. I should have maybe refreshed my memory more on that. Are there any other questions on the chat? Please, if you do have them, please just post there. I did have a question. I saw that you mentioned the Jean-Pierre Pouget was talking about the bystander effect with a range of somewhere between 7 and 35% as an effect of the radionuclide therapy. Does it compare to, how does that compare to your own research in breast cancer?
Yeah, thanks for the question, Jan. With both sort of looking at the cellular level, so I guess I can answer it with two ways. One, at the cellular level, looking at something like at DNA damage, I see sort of like a much larger than, say, that 7 to, what was the higher end of that, like 16%. But I see something maybe on the order of closer to 30 or 40%, which is really quite remarkable. In terms of at the larger level, what we've seen in our mice with a bystander effect, that's kind of currently what our collaborator Didier Rohan of Florida is working on with his Monte Carlo simulation, is trying to sort of ascribe how much of the reduction in tumor burden that we saw was due only to direct irradiation. But it seems as if for one particular breast cancer cell type, a lot of that reduction in the growth. I don't remember the number off the top of my head, but I think it's higher again than Jean-Pierre ascribed. I think it's in the 20% range, was due almost exclusively to the bystander effect.
Thank you. Christopher Lange was pointing out that PSMA is actually FOLH1, so it's far from prostate specific. Right.
Thank you, Christopher. Yeah.
And as I think we are reaching the end of our time, I want to join the many people that actually thank you on the chat and point out how great your summary was. It's difficult in a topic with so many different approaches that, as you demonstrated, has many different isotopes, different strategies, nanoparticles, drugs, and even the DART therapy, to just keep an overview and understand enough to present all of these topics. So thank you very much for that. I think Lindsay has a few concluding words to point out the next seminars coming up. So thank you again, Brian. Excellent review of our workshop. And thank you all for listening.
Yes, thank you both so much. We've really enjoyed this presentation and look forward to working with you both in the future. We also thank our attendees and invite you to join us Wednesday, June 3rd for a complimentary presentation by Drs. Charles Lamoli and Marie-Catherine Bourgeois on flash radiotherapy, what we know and where we need to go. Details and are available at radres.org. And the Radiation Research Society is dedicated to providing education to our community. We have several discussion forums open at this time, including the Red Women in Science, Early Career Investigators, Coronavirus, and coming soon, the RadRes that. You can also look forward to webinar recordings and podcast interviews on our YouTube channel, or keep up with the latest news in the field by following us on Facebook, Twitter, or LinkedIn. And last but certainly not least is our wonderful and always updating education center, featuring courses, books, articles, and additional learning and teaching material. So we want to thank you all again, and Mr. Canter and Dr. Schumann very much for attending, and we hope to see you June 3rd.
Thank you, Lindsay.
Thank you all for joining.
Thank you, everyone.
Thank you. Bye.
Bye.